So thanks a lot, and thanks for the for the invitation, and yeah, very happy to hear. Tell a bit what we are doing at AstraZeneca around machine learning, AI, and of course we apply it in a lot of different fields. But I will focus here on what we are doing around drug design. But first I have some general slides, just introducing the company and what we are doing, our purpose. So basically the purpose with a pharmaceutical company is transforming the future of the healthcare. How can we do innovation? How can we do the next generation of science to deliver the next generation of therapeutics? So clearly there's a lot of successes historically developing new drugs, but there's still tremendously unmet medical need, both say in cancer but also in chronic diseases. And how can we actually use machine learning and AI to basically to find the new treatments. Just some numbers essentially to highlight the size what we are talking about. So we have a revenue close to $60 billion. We have R&D, research and development, investment in close to $15 billion, which is clearly a big sum, and big sums are needed, particularly for expensive clinical trials. And we are also now close to 100,000 employees. So we're talking about it's a big company. I think it's on the Stockholm Stock Exchange, and I think we are 3 times more worth than the closest company. So we are worth around $300 billion. In Sweden, we are located in 3 places. So we have the marketing company in Stockholm, and then we have a manufacturing site, which is one of the biggest in the world, in Södertälje, and then we have R&D in Gothenburg. So in Gothenburg, we are focusing on chronic diseases, so what's called cardiovascular, renal, and metabolism diseases, and respiratory and immunological diseases. And we mainly work on what's called small molecules, so we don't work on biological molecules like antibodies. Numbers here. So in Sweden, we invest in R&D 33 billion Swedish crowns, and basically what is manufactured in Sweden is selling for over 200 billion Swedish crown worldwide. And actually 5% of the total Swedish export is from AstraZeneca. That's mainly from Södertälje, since it's actually one of the biggest pharmaceutical manufacturing center in the world. In Sweden, we are close to 8,000 people. Of them, 3,000 are here in Gothenburg, or actually if you're from Gothenburg, you know we are not in Gothenburg, we are in Mölndal, but Gothenburg is close enough. And then close to 5,000 people in Södertälje. And so what is the purpose? Why are we so eager to apply machine learning and AI? And of course, machine learning and AI is a technology, and you need to apply it for a purpose. And 2023, we set up our ambitious goals to what we'd like to reach by 2030, where of course AI will be one of the key pillar. One is to deliver 20 new medicines. I think we delivered 9 to the market now, and we are well on our way to reach 20 new medicines by 2030. Also leading sustainable healthcare, both in healthcare equity, so our medicines get access all over the world, but also that we apply manufacturing processes that are as efficient as possible to reduce our carbon footprint. And also to be, when it comes to turnover, an 80 billion company by 2030. So that is basically, these are the main goals why we are applying machine learning and AI to reach these. And if you drill down a little bit, why we try to reach that? So how have we tried to do it, and what are the most important factors there? So one is acceleration, and that's what I will talk about here. It is how can we develop drugs faster? how can we reach the patient faster? So acceleration is much more an important key point for us than just to say saving money. Another important part is how can we find mechanisms, so-called drug targets, that works. So how can we apply machine learning AI to find the best mechanism to develop new medicines? So there's really about the quality. And the third is, now we're generating a lot of data. How can we really transform into truly personalized healthcare? And I think there, particularly in oncology, how can we give the right treatment to the patient based on the sequencing the tumors? So really AI is coming in all these steps, and they are really important for us. And we see this is really as a growth driver. And I like to make historical, look at the history to understand why we are now. And one way of looking at it is actually how have we developed drugs over the years. And I think there have been four different eras. And the first era was up to 1950s. And how you developed new drugs then was essentially a chemist synthesizing a compound and then testing on themselves. And of course, that's a way of developing drug. It might not be the most efficient one, but it can work. And if you go to the dentist, you most likely, as a local anesthetics, get lidocaine. And it was actually discovered in that way. So it's fully possible. Then through the 50s, say to the late 80s, early 90s, you basically develop drugs mainly through animal models. So you optimize your compound for therapeutic efficacy in an animal model. And that works as well. One example is Losec, that was developed by us. And it was a best-selling drug in the 1990s. It's for ulcer. But it also have limitation. And in the late 80s, early 90s, it come the molecular biology revolution. So you could actually much better treat individual proteins and individual cell system. So we started to develop new drugs in basically screening direct on proteins, screening on cellular system. And I think we are still in that paradigm. But now we really see a paradigm shift over to AI and computer to drive, so to say, the design. And it's not over a day. I think we are not fully there yet, but we are on our way to basically apply a machine learning AI much better. And of course, for that you need certain input, certain starting points, and one is, of course, the hardware. So I think that's one reason it goes much faster now. We have much better hardware, and I'm really, really enthusiastic about the new spherical high-performance computer that we will get in Sweden. Anders Zinnerman mentioned it in his keynote, and we are really eager to start to use it, and I think we can really use it efficiently from day one. Then you have algorithms. I mean, new algorithms are developing, and I think they're really key as well. But you also, the soft part is really important, the AI talent, to train existing people, to bring in new people on board that have the right competence is extremely important for us. And then also to change the culture, to have more on what's called an AI-first culture, that you think up front in a project how you can generate the data sets that you then can learn from. So overall, you can accelerate your project so you can reach your clinical candidates faster. And then, of course, data, and I will mention that in a few slides. Data is very important for us. For many reasons. One reason is competitive. There's a lot of biotech starting up that is have like an AI-first approach, but I think we have a competitive advantage compared to them because we have all our historical data that we can make use of. So say another slide just emphasizing that when we apply data, digital, and AI to different parts, I'll come back to accelerating drug discovery and design, but it's also transforming how we are working. in the end, many times technology is the easiest part. The most difficult part is to change your business processes. How do you change your business processes? So you set AI first. How do you change your review processes, your governance processes, so you can actually accelerate the whole project, not just some parts with AI? Another, of course, what is important is empower the patients. I mean, now when we basically know much more, we can build models. We do believe we can recommend much more efficient medicines that are more tailored to the patients. And the last part is, of course, extremely important for us, is to work ethically, because we're working a lot with human sample, clinical trials, and you can read on our homepage statements about AI and ethics, or ethical principles, and that's paramount what we are doing to put the integrity of the patient, the ethics forefront. Just a slide also around data, so that's really important. And just to emphasize, we're working a lot on connecting our data and to be able to use it in large language models and so on. Data in isolation many times are not that useful. So it's really to connect the data so you can learn, data types can learn from each other. It's called multimodal modeling before. You should call foundational models now. And that's very important. Going more into my area is called drug design. And it's basically to answer two questions in a drug discovery project. Which compound should you synthesize, and how should you synthesize it? You're in a project, you have a lot of data, but to optimize it further to find the clinical candidate, this is usually the two key questions that you try to get right and try to improve in machine learning and AI. So basically, how is the process working? How do we find new clinical candidates that we can then put into the clinical studies? And if we start with a hypothesis, if we modulate a protein, then we can get a therapeutic effect. And then we need to find a starting point. We can find it through a technique called virtual screening. I'll come back to that. But also a lot of experimental techniques to find your starting point. And then when you have your starting point, you basically you optimize it iteratively. You synthesize compounds, you test them, and then usually they're a bit better, but not really good. And then you move around in what's called the design, make, test, analyze cycle. And then hopefully in the end, you have your clinical candidate. And then you have a clinical candidate that is safe, have efficacy, and is ready for put into the human clinical trials. So this is an iterative process, and it can take up to 4 or 5 years, depending on your starting point. And we, like all other pharmaceutical companies, have an explicit goal. How can we shorten this time with machine learning and AI? Coming back to how do you find starting points, and as you might remember, there's a Nobel Prize given to DeepMind 2 or 3 years ago through innovation, a technique called AlphaFolds. You could actually predict the protein structures just from the sequence. Fantastic achievement, fantastic science. And now DeepMind and also other players, both companies and academia, take it to the next step, what's called co-folding. So you predict not only protein structure, you predict actually how a ligand fits into that protein structure. And that's really a science that advanced very quickly. I don't think we are fully there yet. I think a problem is that you get a static prediction of the protein structure. We are working internally doing research. How can you actually introduce more dynamics so it really better mimics the protein in a cell. But it goes forward very quickly, and this will be one of the application on the spherical machine. How can we scale it up? How can we do this co-folding prediction of a protein binding to a ligand to really large data sets through the spherical computer? Going back to the what's called the design, make, test, analyze cycle. So how can we speed it up? You can speed it up basically in two ways. You can learn as much as possible in each cycle, which means that you reduce the number of cycles. And you can actually select the most efficient routes to synthesize and test your compound. So each cycle you speed up. And of course, we are working on both to basically design the right compounds to learn the most in each cycle, but also combine AI with automation. So you can not only come up with an idea, you can actually test the idea as quickly as possible. And then, so say, why can we do that? And why has happened that we can work in this way that we could not do for 10 years ago? So one, as I mentioned, is the increased computational power. Another is advancement in particular neural network algorithms. So usually in our field, we don't develop any new algorithms from scratch. We borrow from other fields. Particularly, we are using the Monte Carlo tree search algorithm. It's actually from DeepMind, the Go program. We use a lot of ideas from natural language processing. I will come back to that. And also from image processing. So we tag on to other fields and really apply it to our field. And of course, we're using a lot of open source software. And we also try to really contribute back to the field by releasing our software as open source. And of course, we use like PyTorch particularly a lot. We also use scikit-learn a lot, and also specific cheminformatics package called RDKit. And then, of course, what's important is data. And data, both the volume, but also the quality, to find the right combination of volume and quality is very important. And what we're using a lot is, of course, neural networks. So this is, of course, a basic slide. And there's a lot of variants of neural networks. And when I started in the industry, we did use neural networks. But then for quite a few years, we stopped using it. And we used methods like support vector machine and random forest because they were a bit more efficient than our tools. So if you have a molecular structure, can you predict the solubility? Can you predict the permeability? And so on. So for quite a long time we didn't use it, but then it came back. And it came back a lot because it's so versatile, and particularly algorithms that are on the bottom right have been very crucial for us, where you have neural network algorithms where you can learn not only from the input over to the output, but also in the hidden layer there is information going in between the hidden layers, which means it's very useful to use for so to say strings and sentences and time-dependent phenomena. And I'll come back why it's very important for us, because for molecules we have something called SMILES. So it's a language. And you can describe a molecules in a very many different way. On the top, you have a very natural way of describing a molecule. It's a graph. And you can visually understand how a molecule looks like, how the bonds look like, which atoms are connected through bonds and so on. But it's not a very efficient language. So in the 80s, there was a language called SMILES developed that basically described the molecule as a string. And you can see through the color coding which atom in the graph corresponds to which, so to say, letter in the string. And that's a very efficient way of describing a molecule. And it's a language in its own. It has its grammar, trivial to English or Swedish, say, but it's still a grammar, which means that we can apply methods from natural language processing to drug design. So we can see, like when we synthesize a molecule, it's basically a transformation from building blocks to the products. We can see that as a language translation. We can also see the optimization of a molecule as a language translation from the starting molecule to the finished molecule. and I think we can see like exploring the chemical space. So I think it's estimated that there exists about 10 power of 60 molecules. We can essentially use text generation techniques to explore the chemical space. And I will give an example of that. So we have a neural network. We usually use transformer now, but this is a recurrent neural network. But I think that illustrates the principle very well. You train a recurrent neural network or transformer with maybe 1 million compounds, could be from our internal collection, could be from public sources, and then you sample from that trained network. And I will show here, so you have the probability distribution for each letter in a smile string. So the darkness illustrates what is the probability for to sample it. So if it's a capital C, which is an aliphatic carbon, you have the highest probability. Then the second highest probability is the capital O, which is oxygen, aliphatic oxygen. And then you can sample from that distribution. You sample something. You sample the second most probable, the capital O. And to the right, you will see a red ring symbolizing an oxygen atom. And then the trick here, which builds on, is what's called a conditional probability. If you sample your first character, then you have a conditional probability. What will the second character be? And if you check now, we select first as an oxygen. The most probable second character is an equal sign. And that's because in chemistry you have a very common functional group called the carbonyl group. So then let's see which it selects, and then you will see a structure build on the right. So it will select character after character, and then on the right a molecule is built up, perfectly what's called drug-like molecule. So this is a lot what we're actually doing here is taking advantage of recurrent neural network to basically generate drug-like molecules. And through that, we can sample through the whole chemical space. So that's a very efficient way to find new ideas for projects. When you have a problem, you can generate new molecular ideas. But that doesn't solve a problem in a project, because the chemical space is very big, 10 power of 60 molecules, so you need to find an algorithm to actually sample the chemical space. And there we're using something called reinforcement learning. And I think that's a very efficient way of sampling the chemical space. I'll show a toy example here. So you have a molecule up right corner. It's a drug on the market called Celecoxib, and we want to share similar molecule, to find similar molecules in the chemical space. And then we use the RNN to sample randomly in the chemical space. Now it starts to generate randomly molecules. And then through reinforcement learning, when it finds a molecule that is slightly similar to that one, you can see the similarity index will start to go up, and then it goes up like more or less exponentially or sigmoidal, because it learns how to generate molecules that are similar to a target molecule. And that's a very efficient way to sample the chemical space. And that's a research field. So we're doing actually a lot of research within that. And I see Hampus sitting there, just finished his PhD around reinforcement learning for molecular design. Actually was funded. So I'm really grateful for Anders that gave the introduction talk that we got funding for this really exciting projects. And so now actually we can sample the chemical space, and then we need to score the molecules in a proper way. You can find new ideas, but the trick is also how can you score it? How can you assess which molecules are good, which molecules are not good? And then you can do in different ways. And I will show another video here, but it's kind of realistic how you do a lot of work in a project. You have a protein pocket there, and then you try to find molecules that fits into that protein project. You use a technique called docking. And now you generate molecules, and each dot is that it actually fits in. And you can also see the scoring, how well they dock in as well as on the left side. But you can see through reinforcement learning, you can find better and better compounds over time. And that's really a standard way of using reinforcement learning, and something each computational chemist apply to all of our drug discovery projects within AstraZeneca, both here in Gothenburg, but also in oncology in the UK and US. So now we have really found a way to find new starting points. Another interesting way that I'd like to bring up is how we're working. Because, I mean, in a big company, everything is distributed. You need to work so to say, beyond your department. You need to work cross-functionally. And I think we have managed to do that very efficiently. So all the research is done within my department. So we are responsible for all the back end. And then we are working with a business reference group where the user of the tool can give fast and efficient feedback. What is missing? Which feature should be prioritized, what needs to be included in the next version. And then we have an IT organization that's then responsible for the front end. So you work as a team, cross-functional. I think that's very important for AI applications in larger companies, that you can do that. And also to find diverse solution is important. So reinforcement learning is very efficient, but you can find a local minima, and then you get stuck there. So we worked a lot in how can we actually penalize to be in one minima, so it actually can move over and generate molecules from another minima. This has been one of our active research projects here. And here, just to show how efficient reinforcement learning is, it is here you first want to generate molecules that looks normal, and then you say just maximize the number of rings in the molecule. You get actually a molecule consisting only of rings, and then you say reverse and give me molecules with no rings, and then it learns that really quickly, and then you get molecules with no rings at all. So reinforcement is really an efficient technique to sample the chemical space. What is also important is the human input for two reasons. One reason is that there's a lot of implicit knowledge in the human around the drug design that has been captured for the whole career of the drug design of the chemist, and that's not really captured well in any document, so that the human can get feedback if they want to change something is very important. Another aspect that makes it important is that, so to say, we need to create intellectual property patents. And in most jurisdictions, an AI cannot be an inventor. So I think it's really important that we can show that the human makes contribution here. So the inventive step is from the human. So that's very important from an intellectual property perspective. Going back to basically scoring, so this is a quite complex slide. What I would like to say here is that AI machine learning is good, but many times we actually combine it with more traditional methods, what's called molecular dynamic simulations, that we use physics-based theory simulations to estimate properties, particularly binding affinity. Many times are better estimated from physics-based method versus from machine learning and AI. So for us, it's really, we want to be digital, we want to use machine learning and AI, but also combine quite a lot with physics-based methods. Just to say about the reinvent, so that is the novo drug design tool that I showed an example for before. It's it's well published, and we also show success cases where we can accelerate drug discovery projects with using reinvent, where it can be very efficient to find new good compounds efficiently. And also on the bottom, you see the link to our GitHub page. So we publish essentially all our methods and also release the source code. So if you're interested, try it out. Just go to Molecular AI on GitHub and download the code and start to generate your own compounds. It's easy. So the next step, so that has been what we work on several years. What we are now moving into is to do it more efficiently and to learn from the fusion model, but particularly from flow matching. So flow matching and diffusion model have been very successful in image generation, where you take an image and you go to noise, and from the noise you go back to the image. And it would be really nice if you can do something similar for molecules. So you have a protein pocket, and then can you actually generate from noise, from random position atoms, actually drug-like molecules in the pocket. And that's a very active research field, so we're working both with Chalmers and KTH there, and I think we've made a lot of progress. So we have, I think a movie should be starting. I don't know if it's possible to start it by, okay, it starts now. Okay, so you can see basically from random position atoms, a fully drug-like molecules is created in the pocket, and that I think how we will work in the future, that we will basically give a sequence that first it will predict 3D protein structure, and then it will generate molecules directly in the pocket. And that's also why I'm excited what Anders mentioned around the spherical collaboration, because there we will really have an access to a really large-scale computer, so we can take all our protein ligand crystal structure data that we generate for 30 years and actually train a really good flow matching model for molecular generation. So that's something I really look forward with the spherical collaboration. Just going forward, and just to say, we work on small molecules, but we also extend to other modalities. They're a bit more tricky. Peptides is the closest modality. So we have worked a lot in developing methods both for generating peptides, but also for scoring, for assessing that general peptides are suitable or reasonable for your projects. And it's called PepInvent. And then, so to say, what we work really hard on now is also that we have worked a lot on design, but how can we actually move it from more human-based design and particularly human-based experiments into more autonomous design, so we can more have a system that we can actually run 24/7. So then would of course mean that we can develop the drugs much quicker. So go more over to autonomous design, so the labs can be used essentially 24/7, that they're dark labs in the weekends, in the nights, and so on. Is something, of course, we look into a lot. This is one of our labs, is what's called iLab, the automatic synthesis platform at AstraZeneca, which we are using to speed up the synthesis of chemical compounds. And for that, it's of course very important also to move into agents. Like Anders mentioned, and also you can read a lot in the press, agents are very important for basically all AI application. Going over, there's probably no exact definition of an agent, just like AI. I mean, it's a little bit in the eye of the beholder, but it's something around to have an autonomous system that can perceive its environment and somehow make some type of decision and action with a specific goal. And I think that can open up a lot of new opportunities. Agents is actually a very old concept. I think if you read in an AI textbook from the 1960s, you can read about agents. But I think what's made them very sort of powerful is large language models. As you know, There's large language models out there. They get impressively performance over time, and this we can all expect that it will be even more powerful in the future. And that's for a lot of different tasks. We have developed our own agents in the team for chemistry. It's called the Chat Invent, and it's at this stage more of an assistant. I don't say it's that advanced assistant, but it can help chemists to answer simple question in a drug design project without going to look up in databases and so on. If you have a molecule, how can you change it to get smaller, bigger, more lipophilic, less lipophilic, and so on? So you have a question, you go to a supervisor agent, which is an LLM, and that agent goes then to other agents, specialized agents, so get back a bespoke answer from those agents that usually work with underlying tool, and then you get out an answer. And the system called Chat Invent, it was just published in Drug Discovery Today, which I can recommend. It's more, Drug Discovery Today is more lightweight journal, and you can read about Chat Invent there. So basically, Chat Invent have, through the LLMs, access to a lot of tools, and the tools then can be used for drug design to predict synthetic route, do a data search in all the chemical databases, and also predict different types of properties. So I think it works very well on an assistant level, but I think given how fast LLMs are evolving, we believe, and I think that's good reason to believe, we will go from an agent into a more collaborator scenario within the next years. So in a drug discovery project, you will have a collaborator that can actually propose its own ideas. Have you thought about this? Have you missed this? And so on. So I think that's very important. So if we go more and more towards autonomy, and I think that's a very interesting development. And where we like to apply it is, one is how can we combine it with automation? How can we make our labs more 24/7? So how can basically agents create new experimental protocols on the fly, given access to our historical experimental protocols, potentially some other input from humans or from other machine? How can we create the next experimental protocol so we can run the machine with that developed protocol, so it can run overnight and so on? The other is, of course, around the decision making. How can we make the decision process better? There's a huge amount of data generated in genomics, omics, in imaging, in other studies. How can we help the human to make sense of all the data, and potentially how can an agent come up with their own proposal, what should be the next experiment? So a drug discoverer can ask the agent, what are the three most likely or three most optimal experiments to do next? So I think what's important is to have an AI plus, I call it vision for drug design. So it's not only about the AI, it's a lot about the high throughput data generation. How can we generate the right data sets for the AI? Particularly to generate data sets up front, so we can actually run the projects quicker. And the second is it's really the combined with automation. So we not only speed up the idea generation, it's really how can we speed up the whole process? Because if you only speed up the idea generation, really the experimental testing and analysis will take most of the time. So it's really how do you combine it with robotics? It's very important. The third is that it's not only about AI from a computational perspective. We need to be able to do simulations as well. Can be digital twins for larger systems, but it's also to simulate molecules much more efficiently in the future. So particularly to combine molecular dynamic simulation with deep learning, so you can sample or simulate much longer time frames will be very desirable and very important from the future. So here are just some key learnings, what we have learned from working for quite a few years now for AI-based drug design. First, around the generative aspect that I show, that's really a game changer. This is something that we really could not do before. So that's completely novel, and it's something that we really can apply directly. When it comes to synthesis prediction, I've not discussed that much here, but it's a lot around the data generation there. I think we have the right algorithms. Now it's really how can you generate the right data, and that's of course something we're working on. The third is to predict molecular property prediction, and that actually is a field that have moved forward surprisingly slow. And I think that's common not only for us, but in many other fields. When you have structured data you can have in an Excel sheet, it's quite often that methods like random forest support vector machine perform almost as good as your neural network. So I mean, you always need, when you apply an advanced neural network technique and foundational model, you need always to check, can you actually get the same accuracy with a much simpler model? And the last is, so to say, also around the AlphaFold that I mentioned, that was a great innovation, but it's also how can you go further with innovation. You need to further innovate to include dynamics. That's typical. You do a big innovation and then you need to basically step down and look what you get, and then you need to do some follow-up innovation. But of course, I think maybe the most important is not only about the technology. I mean, it's to have this really vision to include high-throughput data generation, automation, and computational model. AI first culture is key, and then continuous training and educational stuff is very important. I just had this slide left. Now I'm running out, but let's take this. So what is the success here for us? And of course I have a lot of success metrics I need to reach with my department. How do we accelerate the project portfolio? But I don't think it's the real measure of the success. I think the real measure is to have an organization where you have trust in your models, that the, so to say, in a project they trust in your individual predictions for molecules, but they also trust if you apply machine learning AI consistently to your project, you will reach the end goal faster than if you don't apply it consistently. I think that's the ultimate goal, not the metrics I have for acceleration of our project portfolio. So with that, I would like to thank myself, and I'm happy to take any question.
Thank you so much, Ola, for sharing so generously. Very, very interesting. Talk about a deep dive. Eh? We have many, many questions. Sadly, we won't be able to answer them all, I think, because we need some lunch as well. But let's go for the first one, which is actually two questions in one. What is your perspective on developing genetic drugs with AI, for example, mRNA or siRNA, where the drug can be identified from the genetic code?
I mean, that's something we're working on. I think there we also apply machine learning AI mainly on a sequence basis. How can we find the right sequences for the antisense oligonucleotide of the siRNA? So definitely we have a team working on it here in Gothenburg. What my team is doing, we try to move into also 3D modeling of the process for siRNA, how it blocks the RNA. That's still quite tricky to do. We are making progress there, but I don't think we have a reliable machine learning model yet. And I hope the increased simulation capacity we will have in the future will help us to really understand the mechanism better there.
Okay, thanks. The next part of the question, how can scientists talk to the genes with AI as an interpreter?
That I didn't really understand.
Can the scientist talk to the genes, like communicate with the genes, and use the AI as
we can. I mean, you can define both down-regulation, which is most common, but potentially up-regulation as well of the regularly part of the gene. So it means designing something that can interact, so the gene is actually performing a specific action. Yes,
okay.
We're working on that.
Could you talk a little bit about the risks of automated chemical construction? Are there any risks? What safeguards are you
I think, well, first, in a way, there's much less risk because you reduce the volumes. So compared to do manual chemistry work, when you go over to automation platform, maybe you have 100 times smaller volume. But of course, all laboratory work have inherent risks, and it's of course important that with inspections and so on, and particularly if you have called flow-based system, they might clog. So it's, I mean, in all this type of work, of course, safety for the people working there is paramount.
Yeah. You were talking about the IP, the intellectual property on drugs. Do you foresee a future where designed drugs can be in public domain? Pharmaceutical companies are rather performing the clinical trials.
I mean, there have been a lot of discussion, of course, about future business models, and I think that's regulatory aspects, and of course also how much society would like to invest in future drugs for Alzheimer's and so on. And that's a bigger question than for me, I would say.
As AI agents gain autonomy, perception and decision-making capabilities in drug design, how should the industry balance the speed advantages of autonomous AI-driven design with the need for human expert oversight and interpretability of AI decisions?
I think interpretability is important, and how I see it, I think it's a lot about accelerate the whole process by giving more choice which compound to put in the clinic. So instead of developing one potential clinical candidate, you develop several, and then you, in the last preclinical studies, you decide which you would like to put into the clinic.
Yeah. Will you ever hold back? I mean, will you see that the development is too fast and the human expert is not keeping the pace?
I mean, you need to do that. I mean, patient safety is paramount here. So you and I think therefore, I mean, there's a lot of tools out there in the wild around agents, for instance, we can't use. So I think that you need to. I mean, AI can only be the advisor here. I think that's very important to say. There need to be a human in the loop, particularly when it comes to closer to the clinic.
Yeah. Can you see a future where there will be completely individualized or personalized medication, or?
I think we are moving into that in oncology a lot with the cell therapy, which is going to be definitely much more individualized. I don't see potentially the same need for chronic diseases.
Okay.
I think that it will be larger population sequence, but definitely in oncology.
I think we need to break there. Thank you so much. Of course, we have a gift for you as well. And this, you saw the umbrella.
Yes.
It's not just any umbrella. It's actually a storm umbrella. So it's very well suited to the city of wind and rain, as we call it.
I do live in Gothenburg, so that can be very handy. Thanks a lot.
Thank you. Big hand, Ola.